There are a number of different hyperpolarization techniques that have been developed in recent years, and a big driver for that has been the prospective application to medical magnetic resonance using magnetic resonance imaging using carbon-13. Parahydrogen-based hyperpolarization is emerging as a competitor to dissolution DNP with certain advantages. And this talk will focus on FIP based on the sidearm hydrogenation method, which was introduced by Ranieri, Bois, and Aim in 2015, and recently reviewed by the has been recently reviewed by the paper at the bottom of the slide. The sidearm hydrogenation method provides a means of incorporating parahydrogen into molecules that are not directly synthesizable as hydrogenation adducts. And the method has been adapted to vinyl and propargyl esters shown in these steps of the process. And it's in principle extendable to any kind of carboxylate species. So if you take a look at that review, you'll see that all of the demonstrations of sidearm hydrogenation have been performed in what could be called a batch mode. That's similar to dissolution DNP in that regard. The hyperpolarized metabolite is administered as a single concentrated bolus, and therefore the administration and image collection must be completed on the timescale of the carbon-13 T1. So this necessitates the use of the fast imaging sequences like flash or echoplanar imaging. So one could envision potential advantages of carrying out that process in a continuous synthesis. And that could, for example, facilitate the studies with lower concentrations that are closer to physiological concentrations, and to look at the steady state metabolite concentrations. It also would allow the imaging to be performed over arbitrarily long durations, which would enable signal averaging, use of conventional MRI sequences. However, that's not without some significant technical challenges. All of these processes now need to be carried out in a manner that's compatible with the flowing liquid, hydrogenation, spin order transfer, hydrolysis, and pH buffering, and then finally the flow extraction into the aqueous buffer. Bubbling is a common method that has been used in many labs over the years. It's probably can be adapted to a continuous flow process, and I think it can be, but perhaps a more efficient method is the use of the AF2400 tube-in-tube hydrogenation reactor, which was introduced by some of the organizers of this meeting, Thomas Theis and Edward. And we also employed the tube-in-tube geometry for the dissolution of hydrogen. And the way that works is you have here's kind of a diagram here. The precursor solution that to be hydrogenated runs through an inner tubing consisting of this amorphous Teflon, which is porous, and it's permeable to hydrogen and other gases, but impermeable to the liquid. And we found this to be, it's automatically compatible with or inherently compatible with a continuous flow hydrogenation, and it's simple, robust, and efficient. So for example, the hydrogenation of vinyl acetate at a flow rate of 1.5 mil per minute, we could see about 70% conversion to ethyl acetate. Now, the next step, we need to consider the precursor design, and that has really been a lot of work done in this area, particularly by the group of Glogler. And there are, examples here showing the vinyl pyruvate precursor structure leading to ethyl pyruvate. And then there's the other two examples shown in this slide, which are propargylic and they offer some advantages in terms of ease of synthesis, faster hydrogenation reaction, and stability. However, the disadvantage is that the parahydrogen nuclei are further away from the carbonyl C13. So this necessitates the use of some clever um, um, isotopic labeling and pulse sequences. For example, the relayed Minerva, where you can take the parahydrogen spin order, put it onto the carbon-13, and then take advantage of the larger coupling between the two carbon-13. So that requires two C13 labels in the same precursor. Um, However, it can be done even without this intervening carbon-13. One can um, simply use the long distance J coupling between the carbonyl C13 and parahydrogen proton in this propylic ester here. And that works. However, one needs to consider the process of this spin order transfer and how um, compatible that could be and how it could be adapted to a flowing process. So also there's some practical considerations. So if you're going to be applying these pulse sequence like Minerva, that is really going to be taking bilinear spin order under weak coupling condition, and you really need a dedicated NMR spectrometer to carry that with double resonance capabilities. The low field technique applied by the Envision team is much simpler, just requires single channel, not even any detection. But on the other hand, small coupling may set some limitation of the residence time inside the RF coil. There's yet another method. This is really not the focus of my talk, but I wanted to bring it to your attention because it is an alternative that uses the intervening methylene group of the probagilic ester to relay magnetization from the parahydrogen uh, protons to the methylene, and then you have a larger coupling, 3.5 hertz, which is maybe better suited for flow process and for faster coherence transfer between the across the ester bond. That happens when you transport the adduct, the propagilic adduct, from low field, like Earth's field, to high field. It happens automatically through a level anti-crossing, homonuclear level anti-crossing in under Altadena conditions. So you could see, for example, that these methylene protons inherit quite a large amount of parahydrogen spin order. And it's due to a level adiabatic passage through level anti-crossing that results in this enhancement. In fact, if you look at this under ideal conditions where you have a strong coupling between parahydrogen protons and a weaker coupling to the third relay proton, then you see that the density operator that you get after cycling the magnetic field from low field to high field is the Altadena density operator. As you can see at the bottom here, this effect depends on the relative ordering of the chemical shifts in a three-proton model. But it has exactly the same form. You get the bilinear spin order, and you get the linear operator terms as well. But when you go through the level A crossing, it's the same operator form, but it involves the third proton. And it turns out that for many of these sidearm precursor, the idealized construct is not too it is a good approximation to these actual systems. And it also, the appearance of the both forms of the spin order enables the use of either inept or Minerva with vinylic or probagilic sidearms with good polarization results. And it is yet another possibility that we had considered for a continuous flow process. Now, it's not the method that we've chosen. We actually are going to go with a much simpler process called magnetic field cycling. It's actually the first sidearm hydrogenation experiment back in 2015 utilized simple form of magnetic field cycling. And that can be optimized by, as shown by the workers by the co-authors of the paper at the bottom of the slide here, that by the careful transport through maintaining adiabatic transport, one can get fairly respectable polarization after optimization. The thing about this is that this method, magnetic field cycling, doesn't require an NMR spectrometer. It's simple. It doesn't and it's compatible, inherently compatible with flow. So that we can focus on subsequent steps, we've adopted on this robust method, and instead of imposing the field time dependence of the field using electromagnet, what we've done is selected the diameter of the tubing. So if you have very narrow tubing like Blue Peak, you'll have very fast, high velocity at a given flow rate through the magnetic shield here. So we go from Earth's field down to, in principle, 10 nanotesla inside, and that occurs over a distance of a few centimeters. So if the flow is high enough, we can approximate the diabatic or non-adiabatic transport into the low field, and then we can adjust the tubing diameter to slow the rate of transport through these level anti-crossings. And there's two of them that occur in allyl acetate, which is the system that I'm showing on the right. So you can see this is under flowing conditions, just by flowing the liquid through this pathway, and we've got like a helical pathway to slow the velocity through this mild gradient at the open end of the magnetic shield. We can see carbon-13 polarization over a period of time here, and this is natural abundant single skin. If you repeat the experiment in deuterated acetone that's been degassed, one can see reasonable enhancement factors. Bear in mind, this is only 1.4 Tesla, but the enhancement factors are roughly 10,000. And we can see carbon-13 signal enhancement of all of the carbons on this allyl acetate adduct. Now, so I think there's really the crux of this whole scheme, this whole process is really the separation of the hyperpolarized carboxylate species. And the way that's done in the literature is using a gravimetric separation. There are a number of limitations of gravimetric separation, speed, and how do you adapt this to a flow process? So this is like, we all use these SEP funnels in organic lab. That's kind of your gravimetric extraction. Another way of doing it, and that's based on differences in density of the liquids that you're separating. Another method utilizes technology based on membrane separation and wetting of a Teflon membrane, a porous Teflon membrane. So molecules that are hydrophobic will wet the membrane, they'll pass through the membrane, and you can separate these two phases. I'm just going to show you a little movie here demonstrating the operation. Let's see if I can get this to work. You see the liquid coming in as a emulsion, and it's separated in real time almost completely in this example because they colored one of the phases. So this technology, it's available commercially from Zyput Flow Technologies, and we compared it with gravimetric separation and showed that you get almost identical efficacy of the separation to gravimetric separation. In this particular case, we use removing extraction of acetone from a 1-to-1 by volume water acetone mixture. And you can see the gravimetric and membrane separation are compared here. The advantage of the membrane is that the process takes 13 seconds, including 1 meter of plug flow mixing and about 0.4 milliliter of dead volume in the device, compared to up to 60 seconds for the complete extraction. Now, we did a separation test by multi-stage extraction of a 1-to-1 water acetone mixture. We've moved away from MTBE to anisole, which has shown better efficiency for removing acetone and separation. It has a larger surface tension than MTBE. So we're going to take our mixture and of anisole with the 1-to-1 mixture of water and acetone and pass it through these separations with hydrophobic membranes in the first two stages. So, and then we're going to pass it through a final hydrophilic membrane and then acquire the NMR spectrum. And you can see results without room temperature and the effect of salting, which you could also improve the efficiency, the efficacy of the mixing by adding salt and raising the temperature. We can get down to 0.88 mass percent of acetone in water, which is close to the 5,000 ppm limit for that for acetone, and it's close to the value that is achieved in the Envision separation in their published work. The anisole, residual anisole, is extremely small. You can see the peak here. This is sodium acetate was separated, and here's the acetone peak, and here the anisole peak and the water peak. The separation was done in protonated water. Yeah, finally, let's see. Oh, I skipped a slide. I want to just demonstrate that we've been able to carry out the flow hydrolysis followed by separation, and one can detect the sodium acetate peak here at 1.9 ppm growing in.